DNA is consisted of sugar, a phosphate group, and nitrogen bases. The bases are adenine, thymine, guanine, and cytosine. The adenine and guanine are purines. With this, their structure consists of 2 rings, while thymine and cytosine are pyrimidines with a single ring structure. DNA runs in an antiparallel direction. This is because the parental DNA, otherwise known as the original strand, runs in a 5' to 3' direction. The complementary strand, or daughter DNA, runs in a 3' to 5' direction so it can match up correctly with the parental strand.
DNA replication begins with the enzyme helicase. Helicase goes between the 2 strands of a DNA molecule, breaking the hydrogen bonds between nucleotides and separating and uncoiling the 2 parts of the helix as it goes. This forms a split between the 2 strands of a DNA molecule, beginning at a split called the replication fork. This opens into what is called the replication bubble, or the area between the 2 strands of DNA. This bubble is held open by molecules called single-stranded binding proteins, which bind to the outsides of each separated strand and protect them from being damaged by outside enzymes. The separation of the 2 strands of DNA can sometimes result in the overwinding of the DNA after the replication fork, so the enzyme topoisomerase is required to regulate the twisting of the DNA molecule.
After helicase breaks the hydrogen bonds between the 2 strands and forms the replication fork, we are left with a leading strand and a lagging strand. Our leading strand is the one on top with the green arrow pointing to it, and our lagging strand is the one on the bottom with the pink arrow pointing to it. Now these 2 strands run in different directions. Our leading strand runs 5' to 3', and our lagging strand runs 3' to 5'. Since we can only replicate in the direction of 5' to 3', the leading strand is much easier to replicate. But before we can start any replication, an enzyme called RNA primase comes in and lays down RNA primers. So this little blue circle is RNA primase, and it comes in and lays down just a few, just so we can start the process. So after our primers are laid, an enzyme called DNA polymerase, which is going to be this green circle, can come in and finish adding the rest in. So it comes in and does its thing and finishes adding all the rest. Now as we move on to our lagging strand, we face an issue. DNA polymerase can only copy 5' to 3', and our lagging strand is 3' to 5'. So what happens then is RNA primase comes in and lays down a short segment of primers. This then allows DNA polymerase to work down the strand, so our little green circle, DNA polymerase, works down the strand, but then it has to jump back where the RNA primase has already added more up here, so then it can work back down in the 3' to 5' direction, again, going this way. So these short little segments that RNA primase and DNA polymerase are working on together are called Okazaki fragments. So after all of our nucleotides are matched up, an enzyme called DNA ligase comes in and works its way down the strand, making sure that all of the strands are bonded together. And then it also does
Nucleotide excision repair is a type of repair system for DNA. It starts with a thymine dimer, which is common when exposed to ultraviolet radiation. Nuclease comes in and cuts the damaged area and removes it. The enzyme is supposed to be green, or here is green, and that's the nuclease, and it cuts at this section and this section where the DNA is damaged. DNA polymerase, which here is orange, fills in the missing nucleotides. The new nucleotides are here in the light blue. Finally, ligase comes in and fuses the old and new strands together. As you can see right here, there's no longer a missing strand. After this, the DNA is fully repaired. The primer that gets put down so Ogasaki fragments can occur are removed once the DNA is complete. Here, the primer is in red. When it is lifted, there is a section that is not fully complete. This is called the telomere. What happens is that this is just nonsense DNA that will eventually get cut off, and once DNA replication occurs. The enzyme telomeres comes in and rebuilds this section so it can prolong cutting into vital genes. Mutations can occur in different ways. There are point mutations, which are just substitutions. Within the substitutions, there is missense, which is where the wrong amino acid is added. This can either be an improvement, it also may destroy, or it could even not change the protein's function. There are also nonsense, which just makes a stop protein where it should not, and it makes the protein non-functional. There are also insertions and deletions, which could be very dangerous because it can shift everything and throw off all amino acids.